students, we are going to revise your chapters Sexual Reproduction in Flowering Plants, Chapter 2, Part 1 for the class 12th Biology. Now, every day I will try to represent a video before you so that you can revise before the date of your exams is announced. So let's start with the Sexual Reproduction in Flowering Plants, Part 1. Now, first, what is a flower? When we are talking of flowering plants, we should be knowing that what is a flower. You can say that flower is a modified shoot because it is present on the shoot parts. It develops from the floral primordia, and these floral primordia, you can say these are the tissues which develops into the different things. Primordia develops first into floral buds and then into a flower. So this primordia, first it will form the bud and then the flower. Flower is the reproductive part of a plant, and there are two things. Androecium is known as the male reproductive wall consisting of stamens, and gynoecium is the female reproductive part of flower consisting of carpels. So you can see from the picture also, the stamen consists of two things, anther and filament. This carpel consists of stigma, style, ovary, three things. Then the petals, sepals, green part, and receptor cells. So these are the different parts of a flower. You can revise as you have studied first time in class 8th also. These are the simple parts of a flower. Now next thing is we are going to study about the stamen. What is stamens? It is the androecium also, or you can say male reproductive unit. You can say stamen. Now stamen is divided into two. One is anther and another is the filament. So two parts are there, anther and filament, attached to the thalamus or a petal. Next thing is we are going to study the structure of anther. The structure of anther or microsporangium, you can say, it is bilobed and dithecheous. So when we say it is bilobed, that means two lobes are present. So when we cut it like this, one is this lobe and another is this lobe, and a longitudinal groove separates the theca. So this is the longitudinal groove which is completely separating this part. A cross section of anther is a tetragonal structure consisting of 4 microsporangium. You can see these things. These are the 4 microsporangia, 2 in each lobes, and later 2 microsporangia of each lobe fuses to form the pollen sacs, and inside these are fuse itself to form the pollen sacs through which the pollen grains are generated, which we'll see in the further slides. Now when we talk of structure of anther, and structure of anther, you can say when we study it from inside, a microsporangium is circular and surrounded by 4 layers. This, you can say, these are the 4 microsporangiums. These are the 4 microsporangium. These are the 4 layers. You can say it is epidermis. You can see this epidermis is there. Endothecium. Here it is endothecium. You can see middle layer and tapetum. So these are the 4 layers which are present. Outermost layer protect the pollen and help in dehiscence of the anther to release pollen. So this outer layer breaks when the maturity of pollen grains take place and the pollen grains are released from the anther. Next is the second part of this structure of anther. When we say, in this you can say the main function of tapetum layer, it nourishes the developing pollen grain. So this is the 1 mark question sometime asked in the exams also. When the anther is young, a group of compactly arranged homogeneous cells called sporangious tissue. So this will clarify your definition of sporangious tissue also. Occupies the center of each microsporangium. So you can see from the structure also, the sporangious tissue are the homogeneous cells which are present. Now next thing is you can see microsporogenesis. Now the process of formation of microspores from pollen mother cells through meiosis is called microsporogenesis. That means we are talking of formation of microspores, or you can say pollen grains. The cells of sporangious tissue undergo meiosis to form microspore tetrad arranged in cluster of 4. So this is the process you can see in the lower. As each cell of sporangious tissue has potential to form tetrad, so each cell is PMC. It is called microspore mother cell. That means each sporangious tissue is having the potential. उसके पास इतना potential होता है कि वो tetrad में form कर सकता है. On maturation and dehydration of anther, thus post dissociation, I think this is dehiscence of anther, not dehydration, dissociate and develop into pollen grains. Pollen grains release with dehiscence of anther. So you can say first this is the diploid 2n nucleus, meiosis 1 divided into 2 equal number, then meiosis 2 haploid number of 4, and this is how the microspore 4 haploid pollen grains or microspores are formed at the end. Next is the structure of pollen grain. When we say pollen grain, male gametophyte, it is also known as male gametophyte in case of flowers. The shape is spherical in shape. You can see from the diagram also. It have two layers wall outward, exine and the inner is intine. This is exine and the inner layer is intine. So this exine is made up of sporopollen, resistance to organic matter, withstand high temperature, acids, alkalis and enzymes, because of which it is able to survive the harsh conditions, and it has prominent apertures called germ pore where the sporulin is absent. So this is the portion through which the entry of this pollen grain gametes comes out and the pollen tube formation takes place, and it's a prominent called germ pore where the sporulin is absent. Intine is thin continuous layer made up of cellulose and pectin. So these are the two substances through which it is made. Next thing is you have to remember this pollen grain cytoplasm is surrounded by plasma membrane, and mature pollen grain has two cells, one is vegetative and the another is generative. So this is the vegetative and the another is generative cells. So the generative cell is small, spindle shaped with dense cytoplasm, and nucleus floats in vegetative cell cytoplasm. You can see from the figure. In 60% species, pollen grains are shed in 2-cell stage, whereas 40% species it is shed in 3-cell stage, in which the generative cell divides mitotically into 2 male gametes. So this is the general things which is happening. Vegetative cell is the bigger one, which is abundant in food reserves and have large irregular nucleus, while it is having smaller the generative cells. And in case of 60% of species, you can see it is shed in the 2-cell stage, while in 40% it is shed in 3-cell stage. Now we come to the next part, the female part, which is known as female reproductive unit, also known by the name pistil. So the pistil is the female reproductive unit of a flower. General structure of carpel, you can see 3 things you have to remember: stigma, style and ovule, or you can say ovary also, 4th part. It may be mono, bi, tri or polycarpellary. So these are mono, bi, tri and carpellary means if it is having this 3 things single structure, it will be mono. If such type of 2 structures are there, then it will be bi. If more than 2, tri, and many if structures of carpels are there, then it will be polycarpellary. Then 2 words are there, syncarpious and apocarpious. You can see apocarpious, here the gynoecium consists of 2 or more carpels and all carpels are free. When the carpels are present but they are free, you can see there are the different carpels but they are free. But syncarpious means when the many carpels are there, these are the parts of, you can say, polycarpellary, but they are fused structures. You can see from here they are fused with each other. Each pistil consists of ovary, style and stigma, and the ovary has 1 or more cavities called the locules. Placenta in locules bear ovules inside this, and number of ovules may be 1 or more, depends upon the different flower or the organism, you can say. The next thing is, you can see the structure of ovule, the very most important structure on which the question comes. It also arises from the primordia or on the placenta. The short stalk which attached with the ovule placenta is funicell. So this is the stalk which is attached with the placenta is funicell. The primordia grows into mass of cells forming nucellus. So this is the mass of cells, and this is known as nucellus and the body of ovule. 2 protective covering of nucellus is called integuments. These are the 2 protective covering, you can see, known as integument, except at the tip leaving a small opening called micropyle. So this is the part where through which the pollen tube enters, and here the integument is not there. This is known as the micropyle end. Next is the basal part of ovule is called classa, that is lies opposite to the micropyle end. So the opposite end is known as classa. Cells of nucellus are rich in reserve foods. So the nucleus cells are richer in a single embryo sac or female gametophyte located in the nucleus, which develop to form the megaspore mother cell, which we'll study next. Next is the male female development of female gametophyte, or you can say megasporogenesis. So remember this, it is development of female gametophyte is known as megasporogenesis, and that is microsporogenesis. So you can see one of the nuclear cell in the microbial region is differentiated into mega mother cell. This is the nucleus region, and it is developing into the megaspore mother cell. The cell is larger, contain dense cytoplasm and a prominent nucleus. So you can see the prominent nucleus, and it undergoes haploids. Number of cells are formed. You can see 4 number of megaspore, and out of this 4, 3 will degenerate, and only 1 megaspore become the functional. So this is the functional megaspore is there. The upper 3 will degenerate, then it will go for 2 cell, 4 cell and 8 cell, which we'll see in the next slide also. Clearly we'll understand how this is happening. So the functional megaspore is the first cell of female gametophyte. You can see this is its nucleus undergo mitosis. So it will go mitosis and 2 nucleus embryo will form, and 2 successive mitotic division in each of these 2 nuclei form an 8 nucleate. So 2 successive mitotic division will result in the formation of this 8 nucleated embryo sac. So this is how the female gametophyte is formed, megaspore genesis, and this is the structure which is given in your NCERT also. These are very important. After seeing these slides, you can easily understand these structures also, and try to understand the different parts of Clazolan, what is Clazolan doing, antipodal cells, and like this. So thanks for watching this part 1 of this chapter 2. Do see the part 2 tomorrow evening. Thank you.